Today we are going to study about bacterial cell wall. The bacterial cell wall, mainly gram-positive and gram-negative bacterial cell wall. So let us begin with structures in peptidoglycan. Peptidoglycan, also called as murin. Peptidoglycan is composed of alternating amino sugars, N-acetyl muramic acid and N-acetyl glucosamine. The N-acetyl muramic acid and N-acetyl glucosamine units are connected by beta-1,4 glycosidic bond, and the N-acetyl glucosamine is observed to be continuing linked with another N-acetyl glucosamine with tetrapeptide chain and pentapeptide cross-linking bridge. As a result, a three-dimensional crystal lattice structure is observed, that of peptidoglycan layer. The peptidoglycans are one of the most important sources of D amino acids. Now, let us move to the gram-positive cell wall. The gram-positive cell wall is chiefly composed of peptidoglycan layer. Around 90% of the cell wall is composed of peptidoglycan layer. The multiple thick layers of peptidoglycan are found, which makes around 50 nanometer of thickness. The gram-positive bacterial cell wall have acidic components called teichoic acid. These teichoic acids are embedded in the gram-positive bacterial cell wall. These teichoic acids are covalently bound to muramic acid in the cell wall of the peptidoglycan. The presence of multiple numbers of phosphate groups in the teichoic acid actually gives it the negative charge, and the overall charge of the cell wall is negative as a result. These teichoic acids also function in binding calcium and magnesium ions for eventual transport into the cell. Certain teichoic acids are covalently bound to membrane lipids called lipoteichoic acids. Now, the gram-negative bacterial cell wall. In gram-negative bacterial cell wall, only 10% of the cell wall is peptidoglycan. The cell wall mainly comprises of outer membrane, which is the second lipid layer. Outer membrane, which is the second lipid layer. It is also the lipid bilayer, mainly containing lipids and polysaccharides, and are as such called to be lipopolysaccharide layer. Simply, it is called to be LPS layer as well. It is negatively charged layer, and as a whole, the cell's charge is negative in this case as well. The lipid part of lipopolysaccharide is the lipid A. It is the fatty acid connected to polysaccharide through amine groups. The lipoprotein present in the inner half, of the outer membrane acts as an anchoring material to get attached to the inner peptidoglycan layer. And the peptidoglycan layer is not that thick in this case. It is only around 2 to 6 nanometer in thickness. The outer membrane is around 15 nanometer in thickness, and total thickness of gram-negative cell wall is only around 20 nanometer as a whole. There are certain protein channel proteins which actually channel in between the cell wall for entrance of different materials, the pore size of which is around 1 nanometer only. The diameter is only 1 nanometer around, and it is embedded in the membrane called to be porine proteins, called to be the porine proteins.